Bismillahirrahmanirrahim. Paper C, Cytology, Genetics and Evolution. The topic is from the portion Cytology according to syllabus, Chapter B. Topic Cell Cycle. Cell cycle can be divided into two parts, a period of growth known as interphase and a reproductive period called mitosis. First one is the interphase, the period between the end of mitosis and the initiation of another prophase. Interphase is further divided into subphases. First one is the G1 phase, the growth phase. It is the first and the longest subphase of the interphase. Events that occur in the G1 phase: the newly produced cell grow in size and the synthesis of transfer RNA, messenger RNA, ribosome and several enzymes takes place. Second phase is the S phase or synthesis phase. The replication of DNA occurs during this phase of the interphase. And last one is the G2 phase. In this phase, the preparation takes place for the chromosome condensation and the construction of microtubule framework required for the cell division. Microtubule that is required for the formation of centriole, spindle fiber. The final events in the interphase occur with the replication of centriole and their movement to the either end of the nucleus. After the completion of interphase, the next phase is the mitosis or cell division. Mitosis, the type of cell division in which parent cell divide into two daughter cell having the same number of chromosomes as their parent cell have. The diagram showing the different steps of the mitosis. Prior to mitosis means this is before the mitosis, the interphase. Each chromosome make an exact copy or duplicate of itself. The chromosome then thickens and coil. Then the mitosis starts, that is the early prophase, in which the centrioles which have already divided forms ester and move apart. They start to move apart from one another. The nuclear membrane start to become disintegrate. Also, the chromatin material will start to become condensed, as a result of which thread-like material or structure appear, which is known as the chromosome. Consists of two parts. These arms are known as the chromatids and this part is known as the centromere. Then comes the late prophase. The centriole and the ester are at the opposite pole now. The nucleolus and nuclear membrane have almost completely disappeared. Then comes the second phase of the mitosis, metaphase. The doubled chromosomes, the chromosome attached to the spindle fiber, line up at the mid cell in metaphase stage. This is also known as the metaphase plate. यानी क्रोमोसोम जो है हमारे पास बिल्कुल सेल के सेंटर में स्पिंडल फाइबर के साथ अटैच हो जाते हैं और ये बिल्कुल जो सेंटर में अरेंज हुए हैं इसको मेटाफेज प्लेट भी कहते हैं. देन कम्स दी एनाफेज. इन अर्ली एनाफेज द सेंट्रोमेयर स्प्लिट, यानी क्रोमोसोम जो है उनमें डिवीजन हो गई है. हाफ द क्रोमोसोम मूव टू वन पोल एंड हाफ टू द अदर. देन कम्स द लेट एनाफेज. द क्रोमोसोम हैव ऑलमोस्ट रीच देयर रिस्पेक्टिव पोल्स. एक पोल ये है हमारे पास, सेकेंड ये है. द सेल मेम्ब्रेन begins to pitch and the center means ये slightly inward movement start कर देती है या इसको furrowing of cell membrane cytoplasm भी कहते हैं. Furrowing means inward movement. The cell membrane complete construction in the telophase. Telophase stage में cell membrane complete जो है अंदर की तरफ move कर जाती है जिसकी वजह से cell दो में divide हो जाएगा. Nuclear membrane again form around the separated chromosome and this is the formation of the nuclear membrane. At mitosis completion, there are two cells with the same structure and same number of chromosomes as their parent cell have. In the case of animals, as the outer layer is cell membrane, so cell membrane हमारे पास inward movement show कर सकती है या furrowing of cell membrane cytoplasm हो सकता है. But in the case of plant cell, as the outer layer is cell wall hard और rigid structure है, वो ये inward movement नहीं show करता. तो इस plant cell में हमारे पास इसके बजाय सेंटर में हमारे पास सेल प्लेट बनती है, सेल प्लेट और फ्रेगमोप्लास्ट. उसकी वजह से हमारे पास सेल टू में डिवाइड होगा. सिग्निफिकेंस ऑफ द माइटोसिस. इट सर्व्स टू प्रिजर्व द कांस्टेंट नंबर ऑफ क्रोमोसोम. It helps in the growth of organism. Because of the mitosis, the number of cell increases, so size increases. It helps in the regeneration and healing. The second type of cell division, that is the meiosis. It is the type of cell division in which the parent cell divide into 4 daughter cells receiving half the number of chromosomes as their parent cell have. Meiosis complete in 2 stages, meiosis 1 and meiosis 2. Each of these 2 stages consist of 4 stages, that is prophase, metaphase, anaphase and telophase. The prophase 1 is the longest phase, further divided into 5 subphases. First one is the leptotene. It is the stage in which the chromosome appear as long, slender, thread-like structure. Slender mean thin. This long, slender and thread-like structure, which is known as the chromosome. The homologous chromosome can be identified by the similarity in the shape, size and the position of the centromere. Second stage is the zygotene. The homologous chromosome come close to one another, as a result of which pairing occur. This pairing of homologous chromosome is known as the synapsis, as a result of which the structure formed is known as bivalent or tetrad. Third stage is the pegitine, in which the crossing over occur. What is crossing over? The exchange of segment between the non-sister chromatids of homologous chromosome, that is the exchange of segment. The point where exchange occur is known as the chiasmata. Then the diploidine stage, ये हमारे पास जो क्रोमोसोम इस हद तक करीब आए हुए हैं, they start to become separated. They only remain attached at a single point of the chiasmata where crossing over occur. यानी क्रोमोसोम जो दूसरे करीब आए हैं उनकी सेपरेशन स्टार्ट होने लगती है. लास्ट इज द डायकाइनेसिस. इन द डायकाइनेसिस shortening of chromosome occur, number of chiasmata begin to reduce and the separation of homologous chromosome completes, but still they remain attached at their tips. These are the 5 stages of prophase 1. Also in the diakinesis stage, the nuclear membrane almost completely start to become Next, the metaphase stage, the spindle fiber formed, the chromosome attach themselves at the center on the spindle fiber. Then the anaphase stage, the chromosomes start to move toward the opposite pole, and in the telophase stage, the furrowing of cell membrane or inward movement of cell membrane occur. The nuclear membrane and nucleolus start to appear again, and the cell divide into 2. This is the meiosis 1. These 2 cells will enter in the second stage, meiosis 2. Again, prophase 2. The nuclear membrane and the nucleolus start to disappear. In the metaphase stage, spindle fiber formation occur. The chromosome attach themselves in the center of the cell on the spindle fiber known as the metaphase plate. Then the chromosome divide and start to move toward the opposite pole at the anaphase stage, and in the telophase stage, the nuclear membrane and nucleolus start to reappear. Furrowing of cell membrane occur, results in the formation of 2 daughter cells. So as the result of meiosis 2, completely 4 daughter cells are formed, having half the number of chromosomes as their parent cell have. Significance of meiosis. First, it helps in the formation of sex cells, egg and sperm, having half the number of chromosomes as their parent cell have. Another significance, the crossing over takes place in the prophase 1 stage of meiosis. It helps in the genetic variation.